Hello friend, today we will discuss about the packing of DNA into chromosome. First of all, we must know about the what is chromatin, what is chromosome, and what is DNA. So first of all we take DNA. DNA is the polymer of nucleotides, or we can say it nitrogenous bases, and these nitrogenous bases are of two types. One is purine and another is the pyrimidine. We will discuss about the structure of DNA in another video tutorial. Today we will discuss about the DNA packing. So first we must know about the nature of DNA. The nature of DNA, the DNA is negatively charged due to presence of phosphate backbone. So overall this other DNA have negative charge. Now what is chromatin? Chromatin, it is DNA plus protein, and protein may be of two types, either histone protein and non-histone protein. Histone proteins are also known as replication protein because they lose or get separated from the DNA during replication transcription and again assembled with DNA and so on. And some other proteins are known as non-histone proteins which help in assembly, and accessory proteins. Now next we come to chromosome. The chromosome is also DNA plus protein. Then what is the difference between chromatin and chromosome? So if we know about chromatin, is the less condensed form found in G0, or we can say it interphase of cell cycle. If we talk about chromosome, it is found in chromosome is found in metaphase stage of cell cycle. Okay, this is the chromosome, this is chromatin, and this is about the DNA. Then what next? How this DNA control the, let's first of all we take example the chromatin or chromosome is separated from cell, and a enzyme that is known as MNase is given to that chromatin into a test tube, and this MNase is isolated from the Staphylococcus aureus bacteria and named as micrococcal nucleases. Micrococcal nucleases, shortly known as MNase. When this chromatin is treated with this MNase, we found that a octamer and on which DNA is wrapped around. This is another in this manner. Now if we saw this octamer, its length is 6 nanometer, and if we saw this octamer, its width is 11 nanometer. 11 nanometer and 6 nanometer is height. And further studies shows that by the gel electrophoresis, the DNA and the proteins are separated, and then further salting of this protein, we found that there is 4 type of proteins are in this H1, H2, H3 and H4. H1 is a protein with a molecular weight of 23,000 and rich in lysine. The H2A is further up 2 type, H2A and H2B. We will discuss about further what is H2A and H2B. The H2A is a 14,000 and also slightly lysine rich. Similarly, this H2B is 13,800 and it is also lysine rich. The H3 protein have molecular weight of 15,300 and it is arginine rich. And similarly, H4 is 11,300. it is also arginine rich. And if we see that the lysine and arginine both are the basic nature proteins and have positive charge, These are the tails which are lysine and arginine rich, if we saw that. Now, if we calculate after MNase, using the MNase enzyme, we found that we cut this DNA here to here. We found that there is 200 base pair of DNA are coiled around this, and the further study shows that this octamer is made up of a 4 type of protein: H2A, H2B, H3 and H4, and each have 2 units. So it is known as the octamer, as they are now 8. Octamer: H2A, it combined with H3, H2B, it combined with H3, it combined with H2B, and it combined with H2A, like this. So these are the 2 unit, 2 unit, 2 unit and 2. That is an 4 unit octamer. And if we find that the further degradation of this DNA, we found that the actual wrapping of this DNA on this octamer, we found that there is only 146 base pair coiled. However, the number of this coiling is may be varied from species to species. Each may range from 147 base pair to 165 base pair in other mammalian cells, but average we found that it is 146 base pair coiled around this octamer. And the DNA which join the 2 octamers, this DNA, here is another protein is associated with this DNA on which this DNA is coiled, this protein, and this protein is known as H1, which connect the 2 histone, and this DNA which connect these 2 octamers is known as the linker DNA. It is known as the linker DNA. If we found the base pair of this DNA, these DNA linker have 54 base, because it is 200, 146 coiled around this, remaining 54 base pair is linker DNA, which is present from here to here, and here to here is for this. Similarly 54 base pairs. Average अगर जो देखा जाए, if we found that in some other species the linker DNA may varies from 50 to 70 base pairs maximum. Right. So this is the octamer which contain the proteins H1, H2, H3, H4. H2 is of two type, H2A and H2B. So all H2A, H2B, H3 and H4 combined together, producing a cylindrical structure known as the octamer, and on which a 146 base pair are coiled, and a linker histone is also present, which is wrapped around the linker DNA, and the size of linker DNA is 54 base pair. And if we see that the one octamer contains 200 base pair of DNA. Now this, what is the coiling? The DNA coiled around only 2 rounds. How many? 2 rounds. That it is 6 nanometer height. And if we found that previously we studied that the width of DNA is also 2 nanometer. So it coiled around 2 nanometer, then 2 nanometer. What is the left? Only 2 nanometer. So 1 nanometer left on upper side and 1 nanometer is left on lower side, and remaining this is the 4 nanometer on which DNA is coiled. So this repetition, this coiling of DNA on this octamer is known as 11 nanometer fiber. 11 nanometer fiber, and this DNA without coiling is also known as 2 nanometer fiber. If it's coiled with this, like this, then it is known as 11 nanometer fiber. This DNA is further, coiled like this. This is one octamer, this is second, this is third, this is fourth, fifth and sixth. 3 in one side and 3 in one side. So it produces, and if we see that the coiling of DNA on this, like this, and this इस तरीके से जब इसकी coiling हो जाती है इस पर, तो it produced a 30 nanometer fiber. It requires another proteins for producing this, which are known as the non-histone proteins, which coil this like here, ASF, like here protein, histone regulator, known as the histone regulator. Another protein which is known as the ASF. ASF1, anti-silencing function, which is known as the anti-silencing function protein 1. Another protein is required here is the CAF1, which is known as the chromatin assembly protein. These are the some proteins. One more is that is the NAP1, nucleosome assembly protein. these are the some proteins which come here and combine this structure like this 30 nanometer fiber and furthermore condense it. They are all, these proteins are known as the, also here all are known as the histone chaperones protein. It's a special type of packing for histone assembly and formation of the chromosome. These are some proteins which are known as the histone chaperone proteins, and this is the 30 nanometer fiber. If we saw this like this, we come here and we found that this is the 30 nanometer fiber which is in the single scaffold like a rod like structure. It is assembled into a rod like structure, rod like. Then it is known as the solenoid form, and this model is very much used in previous days. That is known as the solenoid model of DNA packing. Further study shows that the pattern of assembling of the 30 nanometer fiber is also zigzag, producing the 4 rows. Then it is known as zigzag form and solenoid. So both forms are found. In some species this form is found, and in some species this zigzag pattern is also found. Now if we see further, that is the DNA with 2 nanometer fiber, it assemble into 11 nanometer fiber on if it coiled around the histone. Then these histones are producing a pillar like structure and packed or producing a 30 nanometer fiber like this, and these 30 nanometer fiber are further converted into 60 nanometer fiber by joining the 30 nanometer plus 30 nanometer, then one more, that is the one more 30 nanometer fiber, that is known as the furthermore 90 nanometer fiber, and this 90 nanometer fiber is further condensed and producing a 300 nanometer fiber, and it have a special protein which is known as a SAR, and these are the some proteins which producing a scaffold. Scaffold mean pillar like structures, and this 90 nanometer fibers have a attachment site. This packing, these 30 nanometer fibers which are packed together have a special 80 rich sequence in DNA, from where a protein known as the SAR can attach to these scaffolds. And like this कि we can, जैसे कि ये एक प्रोटीन, ये हो गया ऐसे प्रोटीन, इस पर ये एक तार निकल रहा है, इस पे ये और इस पे ये, अब यहाँ पर एक ये कॉइलिंग है 30 नैनोमीटर वाली, ये एक छल्ला बन गया, फिर ये एक छल्ला बन गया, फिर ये हो गया, फिर ये हो गया, अब ये यहाँ दूसरा, इस पे ये, इस पे ये और इस पे ये, फिर ये यहाँ कॉइल करेगा, यहाँ कॉइल करेगा, तो ऐसे करके ये कॉइलिंग यहाँ पर होती रहती है, जैसे एक तार फैंसी, जस्ट लाइक ए तार फैंसी। इधर इस तरीके से इधर की तरफ होगा सिमिलर पैटर्न, और इन पर ये डीएनए पैक हो जाएगा एंड इट विल असेंबल इनटू ए क्रोमोसोम। इफ वी सी दैट लाइक दिस ऐसे, and it will produce a structure that is known as chromosome, and where these two arms are meet together, this center is known as the centromere. This is known as the centromere, and here is another protein is attached to this centromere region, which is also have a conjugate sequence, that is known as the kinetochore, which play a very important role during microtubules attachment, cell division, or separation of chromosome. Kinetochore. So this is the chromosome factory. A chromosome, on the basis of this chromosome or chromatin regions, when it is less condensed interface, and this type of structure is found at metaphase. So somewhere the DNA, just like in centromere, the DNA is highly coiled. Similarly, at the edges, the DNA is highly coiled. This highly coiled structure at the center is known as centromere, at the end telomere and end region, that is known as telomere. Center is known as centromere and the end are known as the telomere. And somewhere in middle also there is a hard coiling of DNA is also present. So if we stain this chromosome, for example, we use acetylcarmine and many other dyes, as a Jimsa dye. etc. There is a lot of dyes to stain, and nowadays we find that the regions which are highly coiled, the stain is dark. And it is very difficult to open this coiling. By the RNA polymerase. So such region which is dark stain and very difficult to open is known as transcriptionally inactive, or we can say it as a heterochromatin. And these are the, if it is light stain, easy to open, then it is transcriptionally active, then it is known as euchromatin. Further study shows that this chromatin and euchromatin is of further two types. If we say that euchromatin, it is constitutive. Euchromatin, and second is facultative. Euchromatin. This constitutive euchromatin is always a euchromatin. It always remains as a euchromatin. But the facultative euchromatin, once it was, or it may be in future going to be heterochromatin, or it may be heterochromatin which converted into a euchromatin. So it is an interchangeable form in the various phases of life. That is known as the facultative. Similarly, heterochromatin होगा constitutive. Heterochromatin जो permanently heterochromatin रहेगा, it is centromere and telomere. Other than it also shows the facultative which may be converted into euchromatin into heterochromatin. So this is all about the packing of the DNA into a chromosome. That is known as the nucleosome model. However, there is a lot of proteins I forget to tell you about here. Here this packing, this is the scaffold filler protein known as the SAR. They are the part of the condensing proteins. They are the part of a condensing proteins, or it is they have further many type, like SMC, structural maintenance of chromosome. Structural maintenance of chromosome, etc. Or there is a lot of more proteins. Thank you so much. For more topics, kindly follow our channel and remain associated. Subscribe it. This is all about the DNA.